Hello. Today we are going to be learning about protein synthesis. So, uh, protein synthesis involves RNA. So we've talked about RNA briefly, but here are some key differences between RNA and DNA. So first, actually similarity is they're both nucleic acids, and that means they're both made of nucleotides. DNA is double-stranded or the twisted ladder, where RNA is just single-stranded. So it still twists in the same kind of conformation as DNA does, but there's only one strand or one side of the ladder. In DNA, there is the nitrogen base thymine, but in RNA, instead of thymine, there is the nitrogen base uracil. All the other nitrogen bases, cytosine, guanine, and adenine, those are the same. Also, the side parts of the ladder that make up DNA, that sugar is deoxyribose, but with RNA, it is a different sugar that is known as ribose. So overall, RNA, different sugar, different nitrogen base, and it's single-stranded versus double-stranded DNA. Again, showing you pyrimidines and purines, but this time for RNA. So again, there is no thymine in RNA. Instead, there is a nitrogen-based uracil. Uracil will always find its complementary match to adenine, and guanine will always find its complementary match to cytosine. Definition of protein synthesis. So it's the process by which amino acids are linearly arranged into proteins through the involvement of ribosomal RNA, transfer RNA, messenger RNA, and various enzymes. We're going to kind of use a simplified version of the definition when explaining protein synthesis today. So a gene, this is essentially a section of DNA that is used to make a protein. So it says also that it's a unit of heredity. We're going to talk about heredity in future videos, but for the technical use, which is what we're going to be talking about today, a distinct sequence of nucleotides forming part of a chromosome, which is just another form of DNA, the order of which determines the order of monomers in a polypeptide or nucleic acid molecule, which a cell may synthesize. So essentially, a gene is a sequence of those A, T, C's, and G's, those nitrogen bases, and whatever that sequence says, that is going to make a specific protein, and that protein actually does the thing that the gene essentially instructed to. So for example, hair color, there's a protein for the color that your hair is. Okay? And so to keep your hair the color that it is, that section of DNA or that gene, that sequence is read and used to make a protein to keep your hair the color that it is. There are two main kinds of RNA that we're going to be discussing for protein synthesis. The first is messenger RNA, or mRNA, which is involved in transcription. And then there's transfer RNA, or tRNA, which is involved in translation. They are both RNA. The M and the T are essentially just describing what the RNA is really being used for. So obviously, again, mRNA is in transcription, while the tRNA is in translation. All right, so the first part of protein synthesis is transcription. There's a bunch of different definitions there, but I'm going to read the biochemistry one, which says that it is the process by which genetic information represented by a sequence of DNA, so a gene, so a sequence of DNA nucleotides is copied into newly synthesized molecules of RNA with the DNA serving as a template. So all RNA is just copied or complementary versions of the original DNA template. So how does that start by happening is with an enzyme known as RNA polymerase that unwinds a section of DNA, which again is a gene, and makes complementary RNA nucleotides to the DNA nucleotides read. So in that GIF on the right, you can see that is the that purple blob, that is the RNA polymerase enzyme, and inside it is unzipping and matching up the right nucleotides, and it just moves along the DNA strand until it has completed the mRNA strand it needs to be for that protein. Another important part about transcription is that this whole part happens entirely in the nucleus of eukaryotes, eukaryotic cells. All right, so the second part of protein synthesis is translation. Again, in that definition, I'm just going to read the biology part. So it's the process by which a sequence of nucleotide triplets in a messenger RNA molecule gives rise to a specific sequence of amino acids during synthesis of a polypeptide or protein. So the reason that translation is important is because it basically, so the ribosome, so that's the organelle that makes protein, can actually understand the directions that the DNA is sending. That's why the translation part is important. So, second part of protein synthesis, that mRNA strand was made in the nucleus. It now leaves the nucleus and goes to the cytoplasm, which is where the ribosome is located. Then it's going to go into the ribosome, and then also going into the ribosome is tRNA. All right, and this tRNA carries amino acids that have complementary tRNA nucleotides to the mRNA nucleotides being read by the ribosome. You can see that happening in the GIF in the bottom right. When a tRNA molecule has complementarily matched with the mRNA sequence, the amino acid, so the unit of protein, is dropped off and then used as part of the protein. So again, looking in that GIF in the bottom right, those yellow crosses essentially have the tRNA attached to it, and it also has the amino acid attached to it. So we can see it comes in, drops it off the ribosome. Ribosome builds up those amino acids into some kind of shape or structure for the protein, which is going to determine what that protein is going to be able to do. Introducing this content now. So codons and anticodons. So 3 nucleotides together are a sequence of mRNA. So those are known as codons. Okay, so 3 nucleotides, A, C, U, G, whatever they are, those are codons, 3 of them together. Okay? 3 nucleotide sequences that complement mRNA codons are known as anticodons. So that's what the tRNA is. So mRNA, codons, tRNA, anticodons. So that's what it matches up to. There are codon sequences for starting protein synthesis as well as for stopping protein synthesis. So that's how the RNA polymerase can know where to start and stop unzipping or start and stop making mRNA. Same thing for the ribosome. When it's reading that mRNA strand, it knows where to start and stop making that protein. More information, 3 nucleotides are needed to code for 1 amino acid or 1 unit of protein. There are 20 different kinds of amino acids, and the way those, the different combinations of those amino acids, sequences, formations of those amino acids, they make different proteins with different functions. Now the images on the right are codon charts. The next video is going to be all about how we can use those charts to figure out what amino acids make up a protein and how we can determine that from the code that is presented. And yeah, that will be a cool video, so hope you watch. All right, here are all of the steps of protein synthesis. I've essentially said them all already. I'll just let you pause the video and read those on your own time. It looks and sounds more complicated than it is. I have it broken down into 6 main steps here. And here I have some images to help you understand even further. So the top left image and the bottom right image both essentially are saying the same thing, but in a slightly different way. So again, you have transcription, that RNA polymerase opening up a short section of DNA or a gene. RNA polymerase moves down the DNA, making mRNA nucleotides. Then that mRNA strand is going to leave the nucleus where it meets up with the ribosome. And when it's at the ribosome, tRNA molecules are going to come in and match their anticodon to the mRNA codon. And when that happens, it drops off the amino acid or unit of protein attached to it. And then that protein builds longer and longer and longer. In the bottom right picture, it's into the growing amino acid chain. And that's going to keep happening until that stop sequence is read on the mRNA strand and the protein is finished being made. The GIFs in the top right and the top left. The top right shows both transcription and translation, while the GIF on the bottom left shows just the translation part. But what's interesting about the GIF on the bottom left is that it shows that UGA, so those letters in red, that is an example of a stop codon. So when the ribosome reaches that point, it knows, hey, I need to stop making the protein. So I'm going to take a minute just so that top right GIF can go through one more loop, and then I will end the video. Thank you. Thank you for watching. Please share and subscribe, and remember to spread kindness.